Okay, good afternoon, everyone. My name is Danyang. I'm a PhD student from City University of Hong Kong. My supervisor is Professor Ko Chun-Ki, and the topic I'm going to talk about today is the selective targeting of parallel G-complex structure using LRNA aptamer. So first, sorry. Oh, sorry. Oh, now it's okay. Yeah. Okay, so first let's see some introduction information about the G-quadruplex. G-quadruplex is the secondary structure of DNA and RNA is formed in gene-rich sequence. The guanine in gene-rich sequence can interact with each other to form a G-contact, and two or more G-contacts can interact to form a G-quadruplex. And based on orientation of those strands, it can form parallel, anti-parallel, and hybrid G-quadruplexes. The G-quadruplex structure has been found in different species and also in the different positions of human genome. So it's related to many biological processes like replication, transcription, and translation. So it's also related to many diseases like cancer. The targeting of G-quadruplex is very important. The current targeting method include small molecule ligands, fluorescent probes, and antibodies. The small molecule ligands usually to regulate the G4 mediated processes, and the fluorescent probes and antibodies used to image G-quadruplex. These methods can provide high specificity to G4 over other DNA and I structures like the duplex, single strand, or hairpin. But there are very few methods that can specifically target a certain G4 conformation. As I have mentioned, the G4 has different conformations, the anti-parallel, and hybrid. So the challenge is to targeting specific G4 conformation. And besides this targeting method, another method is aptamer. However, aptamer cannot be developed for nucleic acid structures because in the selection process, the antisense oligos can be selected, and the antisense oligo target the nucleic acid through base pairing instead of the structure recognition. So the LRNA aptamer has been developed to target the nucleic acid structures. The LRNA is the mirror image of DNA. It has the same physical and chemical structures of DNA, but it's highly resistant to nucleus degradation, and it can hybridize with the DNA. So it can be developed for DNA or ddNA targets. The current selection method for LRNA aptamer is the indirect selection method. So before selection, we turn the D target to the L target, and then select a D aptamer from the DI library to target the L target. And finally, when we get a aptamer sequence, we will turn the sequence to LRNA aptamer, so it can target the D nucleic acid targets. Our objective is to develop an LRNA aptamer to target special G-quadruplex conformation. And here the target I use is CK1DG4. It's a 22 G4 sequence in the promoter domain of CK proto-oncogene. And the reason to choose this is because its structure has been solved and it's a standard parallel G-quadruplex. So by using this G4 as a target, we aim to select LRNA aptamer to target parallel G-quadruplex. The method I use is G4 select seq, and as I have mentioned, we use the indirect selection method. So before select, we change the DCK1 target to LCK1 target, and it's labeled with a built-in for later immobilization to base. And we start from a single strand ddNA N40 library, and after a few rounds, we did NGS. We add barcode and linkers and perform the NGS to get the sequence information. Here I want to emphasize that in the previous select method for LRNA aptamer, people use Sanger sequencing, but the Sanger sequencing has very low sampling. So if the non-specific sequence is highly enriched in the library, so it's very hard to find out the binding aptamer aptamers. At first, I also tried a few trials to use Sanger sequencing, but I didn't get a binding aptamer. So later, I modified this select method to use NGS, and finally, I get a binding aptamer. So the whole process is G4 select seek, the sequence analysis, aptamer candidate verification, and the aptamer construct optimization. So now we can see the aptamer. I select some candidates and test the binding, and finally the aptamer 12 provides the strongest binding. So this sequence was selected for further investigation. The first step is to remove the fixed primary region, because the fixed primary region is used for user in the selection process. It's not responsible for binding. So we remove this and get the N40 sequence, the aptamer 12.1. And later I want to make the aptamer shorter and the band stronger. So I optimize this sequence. First, I remove the two flanking sequence. This AGA and the CGA, and later to make it more stronger, I change the UG mismatch to a CG base pair, and to make it shorter, I remove the weak UA base pair, and finally I get the aptamer 12.6, the final aptamer sequence. Next, I want to optimize the loop sequence of this aptamer. Here I use the single nucleotide mutational analysis. I designed based on the purine to purine, pyrimidine to pyrimidine principle, and I designed the 14 mutant sequence, and from then I test the binding. So from the sequence, you can see the original aptamer provides the strongest binding, and all the mutant forms have lower binding. So we keep this sequence, and from this result, we can see most of the G base in loop has large effects on binding. And the next, I optimize the binding cofactor mechanism concentration. When there is no mechanism, the binding is very weak, and when there is 1, 2, 5, 10 millimolar mechanism, the binding gets stronger. And I selected 10 millimolar mechanism for the in vitro binding assays. Then I test the binding using MSAN, and the KD was calculated to be 40.9 nanomolar. Next, I want to see the structure of the aptamer, because the aptamer is quite G-rich and has some G motifs. So we think there might be a G4 structure. Here I use the spectroscopy assays, including fluorescence with THT, fluorescence assay with NMM, and the CD and the UV melting assays. Because the potassium can stabilize G4, but the lithium not. So I set the experiments in both potassium and lithium conditions. So we can see in this assays, the signal in potassium condition is higher than lithium. So they suggest that there is a G4 conformation in the aptamer. And the TM value was tested to be 70 degrees. It's very thermostable. Next, to get more structural information of the aptamer, I also did the structure probing. The lane 1 to 4 is the sequencing lane. I use the DDNTP. I add the DDNTP in the reverse transcription assay, because the DDNTP is lack of two prime hydroxyl group. So when it's added to the DNA product, the reverse transcription will stop and has the stalling here. So by reading this. By reading the sequence from the sequencing lane, we can it's consistent with the aptamer sequence, and this can be used as a ladder. So next, I perform the reverse transcription stop assay in both potassium and the lithium condition. Because in the potassium condition, the G4 can form, so there is a RT stalling. But in the lithium, there is no G4 structure, so there is no stalling. This also suggests the G4 formation in the aptamer. And the next, I want to see after aptamer targeting, how the structure change of the aptamer. Here I use the NAI to modify the RNA base. NAI can modify the nuclear base of single strand RNA, but not G4 or the double strand RNA. So let's see the lane 7 first. So we can see here, here and here are the two stems of the aptamer, because the stem is in double strand, so it has a very low NI reactivity. And this is the loop, but because the loop is in a single, strand, so it should be very high NI reactivity. But we can see some are high, some are lower. This is because there is a G4. I'm sorry. This because there is a G4 conformation, so the G4 can protect the subnuclear base from NAI modification. So some has lower NAI reactivity. And after I add the different concentrations of target, some of the nuclear base has lower NAI reactivity. So like this one, and also this one. And this one. This is because after target binding, the target can further protect the nuclear base from the NAI modification. So the NAI reactivity has some change. So in summary, this assays have suggested the G4 conformation, G4 structure in our aptamer 12.6 and is responsible for binding. So now we have the sequence and have the structural information of aptamer 12.6. Then we convert it to the L aptamer 12.6 and test its binding to the DCT1 DG4. And the KD was calculated to be 42.3 nanomolar. It's similar to the aptamer to L target. And because we aim to select aptamer to target the parallel G4, so next I test the specificity to different conformation of G4 structure. The parallel G4, anti-parallel G4, hybrid G4, and the parallel RG4, because RG4 only falls in parallel G4 structures. And from the results, we can see our L aptamer 12.6 can specifically target parallel DG4 and parallel RG4, but can't target anti-parallel DG4 and hybrid DG4. So suggests the conformation selectivity of L aptamer 12.6. I also tested the specificity to other non-G4 structures, including the G4 mutants, poly A, poly T, poly C, hairpin, D, and RA, and we can see only our targeted CT1D G4 has shift, but none of the other structures should binding to the L aptamer 12.6. And I also tested the aptamer and the target with the same chirality. The D aptamer to D target and L aptamer to L target, so you can see there is no binding. Next, I want to study the applications of L aptamer 12.6, and the first one to study if it can inhibit the primer extension. Here, I designed a template contain G4 structure, and the primer is labeled with the 5' FAM. So in the primer extension process, if the G4 structure form, there will have some stop product, and if the GG4 aptamer or ligand bind to the DG4 in a template, can further stop the primer extension. First, let's see the parallel DG4 sequence template. And when there is no aptamer, there are two kinds of products, the full length product and stop product. And with the increased concentration of L aptamer 6, the full length product gets lower and lower, and finally all the full length product turns to the stop product. But for the mutant DG4 sequence, the full length product didn't change because the mutant DG4 can't band with our aptamer, so the aptamer can't affect the process. And I also designed to the templates with the anti-parallel DG4 and hybrid DG4, and you can see for both of the wild type and the mutant sequence, the aptamer has no effect to the primary extension. This proof that our aptamer can only target the parallel DG4 and affect the primary extension process. And next, we want to see if our aptamer can go to cell and cell nucleus, because the DG4 is in cell nucleus. So here I transfect the FAM labeled L aptamer 12.6 to cell, and use the whole text the HOX to image cell nucleus, and after merge we can see our aptamer can really go to cell nucleus. So next we study in cell application. The first one is the reporter gene assay. Here I use a PSI check to plasmid and insert the wild type and the mutant construct to the HSV2 promoter. This promoter regulates the firefly luciferase. So I transfect the plasmid together with the L aptamer 12.6 to cell, and we can see with increased concentration of L aptamer 12.6, the luciferase activity of firefly gets decreased, but the mutant didn't change. And to see the effect is caused by transcription or translation level, I also perform the qPCR to test the MR level, and we can also see with aptamer addition, the MR level also get decreased. This means that our aptamer can target the CQ the DNA G4 and regulate the transcription process. And we also use a G4 ligand BRCA19 as a positive control. This G4 ligand can bind to G4 and affect the transcription process. We can see the ligand can also achieve this effect, but however, in lower concentration, it has very low effect, and only when we use about very higher concentration than the concentration on an aptamer, it shows the similar effect with our aptamer. This means that our aptamer has stronger effect than BRCA19 in this reproducing assay. And next, I want to see if aptamer can regulate the endogenous gene expression. So here I use the HGG27 cell lines. This cell can express the CK protein, and then I transfect the L-aptamer 12/6 to HGG27 cell, and then use Western blot to test the protein expression. So we can see with increased concentration of L-aptamer 12/6, the CK expression gets decreased, but the control gap DH didn't change. And I also use the PDS as a control. The PDS is the G4 ligand. It can also bind to G4 and regulate the process. So we can see with the same concentration of PDS, the effect is lower than my aptamer. And I also use another LRNA as a negative control. So we can see this LRNA has no effect on the CK expression. And I also test the MR level of the CK of the CK protein, and we can see the MR level also decrease with the aptamer addition. So this suggests that the CK1-DG4 can form in the CK promoter, and with our aptamer addition, the aptamer can target the CK1-DG4 and block the CK expression. In summary, we have developed the G4 select-seq selection method and identified the first L-on aptamer to target the parallel G4 with high affinity and selectivity. Our L-aptamer 12/6 can be a valuable tool for selective recognition of G-quadruplex conformation. And our G4 select-seq can be a general platform for L-on aptamer selection and can also apply to other nuclear acid structure targets. So finally, I would like to thank aptamer 2023 for its oral presentation, and thank my supervisor, Professor Kun Kiko, and all the collab members. Thank you for your attention.